Brings me to one of my sort of major favorite points. Science is communication. It's a communication with nature, but it's also a communication between people. If you believe that the reason for studying computer science is because then you can be completely alone with your computer and you never have to see anybody else in the world. There are careers in computer science where this belief will come true. But I believe it is much, if you want to sort of grow, let's put it like that, then you have to get out there. And out there, doing science is always communicating. It's communicating your science. It's communicating what you find. It's communicating your views and your understanding. This is a very essential part. We cannot today understand really science without this process of communication. And communication often starts with questions. So please interrupt and drive the advance of this lecture by your questions, by being there. Now, I talked about the central dogma, so the information flow. Genes, or the DNA, is translated to proteins. Why do we need this? Why did nature invent genes? I mean, if proteins are the machinery, why not go right to proteins? Why this detour of genes? Why do we have them? Any idea? Yeah?
Maybe they are more stable over time than proteins.
That is one important issue. So genes are more stable in time. So now that brings up another issue. What does it mean, stable in time? No, I mean, most of us do not care if our DNA would be thrown somewhere and it could survive for a century. We care more about how we feel today. And so it's slightly different for our other organisms. You may know that there are a lot of organisms that can stay frozen or dried up for decades or centuries and then come back to life. But now if I just take the air like that and I put DNA or living material in there, is there any danger that something takes it away? How would that living material be endangered in your view? What's in the air that you cannot see, that sort of endangers that living material?
Oxygen?
Oxygen, yes, so that's one important issue. Oxygen, so life started full of oxygen and that was a very, very killing situation. So there's a lot of destruction through oxygen, and yes, proteins would in fact be subjected to oxygen and that would not be good, but there's something else in there. Yeah?
Are living organisms like bacteria?
Lots of living organisms, lots of bacteria. So the air is teeming with bacteria. That's the other thing. So you can actually sort of take a gulp of air and sequence what you find in there. And most of the things you see are novel discoveries. And people have done that again and again. This is full of enzymes. It's full of genetic material that would cut proteins. That would, in fact, that even cuts DNA. But DNA is more long-lived, so that is one of the main reasons. The other main reason we assume is that this is sort of the historic event. Life started with RNA, most likely. So somehow at the beginning of life is a simpler alphabet, is a simpler way of doing this. And RNA found a way to sort of do some of the chemistry it couldn't do, that was necessary for replication, because part of life is to replicate, enslaved amino acids and brought in somehow proteins. That is one way of seeing that. And then sort of a more complex world evolved. Now I said part of this dogma is that the sequence uniquely determines the three-dimensional structure. Okay? Why is this lecture about protein structure prediction? We want to know how proteins function. So when there is a disease, when there's a problem with a protein, typically this is because something goes wrong. And you want to know, what is this protein doing? Why is it going wrong? That has to do with what it does. Why not go directly to function? Why do we care about structure? Exactly. It's as simple as that. It's much easier to handle shape, represent, he said, shape than function. And represent means it begins in the computer. Shape is totally trivial. It's a coordinate in 3D space. And every computer can put that easily. And anybody of you can program this, can read coordinates, can program a database that would contain coordinates. And you could even have little parsers that would rotate these molecules. Function is something much more complex. Just, you cannot put it into numbers. So, structure is related to function. Starting from structure is good, and we can really compare structures. We can look and we can represent structure much easier. I said in the last lecture that sometimes two parts of the same protein. Yes, sorry.
Also about the maybe this shape determines function in biology. Doesn't it isn't it like always shape-driven mechanisms?
So it is true that structure, so getting to the structure is also good because from the structure I learn the function. This is totally true. And shape determines function. This is true. So knowing the shape, I learn a lot about the function. It's not that direct as in when I know the structure, when I see a three-dimensional structure, I know exactly what it does. We are still not able to do that in all cases. When I know only the sequence, it is much more difficult to guess a completely new function than when I know the structure. So the shape is sort of the key to understanding function. The shape is the key to doing the function, but I still don't understand the alphabet or the language, so to speak. So if I could go directly to the function, that would be great. But we cannot do that. Another aspect of structure, and that relates also to what you said, shape determines function. There's not a single drug on the market that is target. Drugs are molecules smaller than proteins. And they always target proteins. And for any drug on the market, one of the first steps is what we talk about today, to do alignments, to do database comparisons. But a little bit further down the pipeline of the drug development, you will have to determine the shape of the protein that you're targeting. There's not a single drug on the market that didn't use bioinformatics, and there's not a single drug on the market that did not sort of use the shape and learn about the details of the shape, such as this image, and beyond that image. Now, as I said, sometimes two copies of the same protein have to act together, and the ADH on this image here is an example for that, in order for ADH to function. So if it's not two of the same homodimer, dimer is two come together, homo means the same. So if not two of the same, in this particular case they are sort of inverted, but if not two of the same come together, then it will not do its function. And there you already see that in order to function, proteins need to interact. They need to form communities. How to get to the shape, and now again, nature does it. We do it by experiments. I already mentioned there's X-ray crystallography. So you use large X-ray beams from large synchrotrons, in CERN for instance. You shoot that at crystals. That means that you have to actually have the protein in the shape of a crystal. You have to express, as it says, so put a lot of the same protein in a small space, and hope or wait until it crystal grows. And that is sort of a science or art in order to do that, but it works for thousands of proteins, which is why we have thousands of structures. I'm going to talk next Thursday, about some ways in which we can predict structure and shape from more or less sequence. But let's begin with a sort of dimensionality or the information content of the problem. On the simplest aspect here, we have 1D, a string of amino acids. That's the sequence. We refer to that as the sequence. On the other hand here, that's a cartoon of a three-dimensional shape. Three-dimensional structure, 3D. So 1D, 3D. In between is 2D, the contacts or the distances. So what you see in this contact map here, you line up a protein. In this particular case, the protein has 1 to 99 residues. So these are the residues in that direction. In that direction, every dot here means that these two residues are close in space. The darker, the closer they are. So essentially they touch. The gray means that they are in the vicinity, but they don't touch. Now, in this distance map, so in terms of information content, secondary structure. So this image here has these arrows. Those are secondary structure. Secondary structure is something that you can see on 3D structures, you see two main secondary structures. One is this helix here, and I will get more details about that. And the other one is this strand. So these are the two sort of main secondary structure segments. Secondary structure itself, you can write as a string. Shown here in this column, where the E stands for extended, that it happens to be this arrow. E, so there's a bunch of E's, a bunch of E's, a bunch of E's, and if there's nothing in this column here, it means it is the third state, none of the above. It's neither helix nor this extended state or this arrow state here. So in that sense, secondary structure is a 1D feature. You can project it onto a string, yes? What is a residue? Let me repeat. So proteins are made up of amino acids. And the moment they are joined in the protein, we refer to them as residues. So one particular position, one particular pearl, if you think about it as a pearl chain, one pearl is a residue. The second question is?
So when you talk of secondary structure, so in mind we must have something which is 3D?
Yes. So when you go to Wikipedia and you would look up secondary structure, or in many textbooks, you would have sort of an image that says there's a primary structure, it's a sequence, a secondary structure that sort of has some idea about the protein structure, a tertiary structure, which is a 3D structure, and then something else. This is a little bit of a misnomer, because in terms of information content, secondary structure is a 1D feature, because you can write it as a string. Anything you can write as a string is 1D in terms of information content, right? The reason why secondary structure forms, maybe you need some information that is not essentially 1D. Maybe you need some information from something that happens somewhere that is not quite in the local vicinity of a particular point in this protein. But that's a different story. As long as you can write secondary structure as a string, it is 1D, okay? So this is a misleading. Textbooks are full of that. It's a misleading idea. In terms of information content, it's very simple. This is 1D, and that is 3D. Secondary structure is a 1D map of a 3D feature, so to speak. Or is that accepted as far, or should I go further? Yes?
I have another question, because last lecture we talked about that you can kind of show the 3D one and the 2D map.
Yes.
So just have a difference from one point to
yes.
the other points. So my question is, if you have like this 2D representation, which is, I think this one is a little bit different in how it kind of represents it, could you kind of transform this one to the 3D map we were describing yesterday?
So the question here is, can we transform, if I had, so this is 3D, this is 2D, so I reduce the information by one dimension. If I did that, and I arrive at this point, can I go back? Can I go from 2D to 3D? And I already mentioned briefly on Tuesday, the answer is yes. As long as this is sort of a complete distance map, that means you are not, so sometimes people talk about contact maps and they talk about distance maps. The difference simply is, in a contact map you only have a binary version here. You have only black and white. In a distance map you have the values in between. You do need the values in between. In order to completely convert this into that one, there is, the only caveat of that story is you cannot distinguish between this image here and its mirror image. Those produce the same contact map, distance map. But other than that, yes, you can completely go back. Now, unfortunately, there is no algorithm that does it in a way that is deterministic. But you can simulate that, and it's essentially an optimization problem, but that optimization problem is on the level of a typical protein, a few hundred residues, relatively simple. Relatively simple translates to your machine can do that not quite in a day, but it's not that simple. But it's something that is really doable. So essentially it is completely doable in the sense that you can do it, full stop. So it's as physicists tend to argue, a trivial problem because it's solved. Yes?
The 2D structure is supposed to be symmetric, right?
3D structures are not symmetric. Symmetric to what?
The 2D, the metric.
Oh, yes, I'm sorry. If you cut off the one diagonal here, you only need one diagonal. So the two diagonals are completely symmetric to each other. In this representation, that's true. Yes. Why is it not?
I guess it's hard.
If it's not, then there's some problem. So I believe that the asymmetry here has to do with time and reproducing. So this is an image that was that I had in my PhD thesis that was in the last century. So went through all kinds of states of display techniques, so to speak. So it should be completely symmetric. Now again, from here, sorry?
It's not symmetric.
So on top there's an arrow, you say? That could also be true, but it should be pretty symmetric. So there's nothing in there that should have made it asymmetric other than time.
And maybe errors. Inaccuracy.
No, because it's plotted from the same data in two directions. So in that sense, okay, let's not go into this image. Let's redefine this. This is symmetric, okay? This is the resolution of my images. I apologize for that. Nobody ever stumbled over that. And it's true. There are tons of examples where we see the lack of symmetry. I take your point. In principle, it should be symmetric. Okay, let's take this as a very good prediction. So the lower diagonal is a prediction of the upper diagonal. And it's not true. And certainly those days we could not get predictions that are anywhere as good as that. We can go from here to there. Can we go from here to there? Can we go from 1D? So we go from 3D to 2D. We reduce by one dimension, but we can go back. So we do not lose essential information. Mirror image we can figure out, so that is not essential. Now we go exactly another dimension further, from 2D to 1D. Can I go from 1D to 3D? And already several people said no. Do you have to say more?
Theoretically, you can say yes if you have a large enough knowledge base. Or
That's a very good point. So he makes this point that I repeat what I said before. I take this chain, I throw it into a solvent adopt. So you need three-dimensional structure. That means the protein knows how to fold. That means in principle, there's nothing else new needed here than the energy of folding. And that means we should be able to do that in the computer. And we still can't do that. Because our understanding of these energy functions is not good enough, because the problem is too complex. There's a variety of problems that are in that way. So from here, we cannot get to there in a practical world. That problem is absolutely not solved. Although the protein, and that ultimately is underneath what you say, is solved in nature. Now, today I want to focus on sequence comparison methods. And so-called alignment methods answer the question of how similar proteins are. Let's begin with this one here. So we have two strings. Say these are two proteins, OPA and PAPI. And in order to compare them, what you will do is you will find the optimal way in which they become most similar. So it's very simple. You sort of move things around until you find some similarity. Let's assume that there is another string, PAPA. And then you keep going, and you create something that people refer to as a multiple sequence alignment. So the alignment, initially the pairwise alignment is the first two, and it becomes a multiple alignment if you sort of bring in the third. And in bringing in the third, what I sort of suggest in this image on the whiteboard now is that we sort of have a group of related things. In terms of the 1D feature, those things are related, and you see that there is a sort of similar difference between the first and the second, and the second and the third, and there is a sort of similarity between them. But in terms of biology, it may not be the same, right? So while I could, if those were protein fragments, I could just align them, the biology of the story may be different. So this one here may mean something totally different. Or not. Well, this one doesn't mean something different. Most of you are aware of that one. But, oops. So you see the father, so the father and the grandfather are sort of intermingled in this alignment. So they don't group the words for father and grandfather. And then there's something else in there that has some word similarity. And in some sense, figuring out what that is, what belongs into this family, that's the part of the biological interpretation. The first part, simply the task of finding the optimal superposition and then computing what this superposition that you get is. And then hope that the higher the similarity is, the more likely you have some sort of biologically meaningful. Right, what is good, what is defined by this objective function. That is defined by what you try to optimize. So what I did on the examples here, simply I optimized the identity of letters, right? There could be other objective functions. And in fact, the simplest objective function is really one of percentage identical letters. All of that, however, still sort of ignores the biological background, the meaning. But let's first forget about that and let's just look at the string matching problem. So the goal is to find the optimal alignment, and optimal for the time being I will say number of identical letters in this match. And I want to sort of find this optimal thing. Now, when I do this, I immediately see that there are essentially two different paths. One we refer to as global and one we refer to as local. Global means I sort of ascertain that these two strings are aligned end to end. So the first of the one is aligned with the first of the other, and somehow the last. The dot here means I put what we refer to as an insertion. And the local alignment would argue, well, I do not necessarily force, this is not a good example, I should change this example. I do not really force that the first one is the first one, but in this particular case, the last one is still the last one. So imagine that, that I would get this out here, and I would only align some local fragment. That is a local alignment. And now, the historical interpretation, or the interpretation of this alignment is really that we have a relation, an evolutionary relation. Why do we care about finding an evolutionary relation? Between two proteins. Why is that relevant? I mean, it's fun, but yeah?
Because the function is probably similar.
That's the idea. So when two species separate, most of the proteins, most of the genes they have are the same. Because yesterday, maybe there's one or two, very, very, very few are different. So when species split, at the point of splitting, they essentially come from two separate populations that are genetically relatively similar. So most of the proteins they have will be very similar. They will do the same thing, okay? So we are sort of tracing history because through tracing history, we essentially believe that what we trace is similarity in function and structure. And that ultimately is what we are after. In this field, often the word homology is used, and this is a slight misnomer. Historically, homologous, homo is agreeing, same, logos is the word, and historically it really comes from this idea that you have different organisms in different creatures. Is coined by Richard Owen in 1843. The same organ in different animals under a variety of form and function. That means that the whale was a mammal, and you still see all the limbs of a mammal in a way, but when you see the whale swim, you don't see the feet. But ultimately, there is an organ of the whale that corresponds to the feet. That is the idea of homology. Now, in this field here, typically homology is applied to the level of genes. So we are not talking about the organs being similar, but the constituents. The constituents here are the genes or the proteins. So we say that people tend to say that two proteins are homologous if they have a similar function and a similar structure. Clearly, this is sort of the underlying thing, and it clearly reminds us of the evolution, and evolution again implies similarity because of historical reasons. And that is in fact what we are trying to go for. One reason why we do this is we compare sequences to find similarity in structure. One reason another reason, essentially, if you found a new sequence or a new protein, you would ask immediately, is there another protein somewhere out there that I know something for, structure, function, or whatever? So you have to sort of compare against the database. The simplest way in which you can do that is a dynamic programming. The dynamic programming essentially does a brute force. It goes back to a paper published in the 70s by Nielman and Wunsch, or in 1970 is the date. And essentially what it does is you take the second, so you begin with the first string, and you try to find the similarity of the second string. The way you do that is you try all possible combinations. You completely slide the second string through. And you simply try all possibilities. You do this through a matrix where you have the first protein, you write the second protein, and then the ones here mark any similarity between the two, identity. And what you essentially do, you try to find a path through this matrix. And essentially the longest path that gives you the highest score is the optimal alignment by tracing it back. Right? It's a very simple algorithm that is giving you relatively quickly a way to do all possible comparisons in a time that is essentially in the square of the length. So if one has the length L1, one protein, the other one L2, then essentially this is L1 times L2, or essentially L square, and slightly faster than that, yes?
Does it make sense to not punish the algorithm at all if it aligns a lot of things which kind of don't at all, kind of?
So would it make sense, I believe what you're saying is, could we mask out regions that cannot be aligned at all? Well, if you sort of first go through the matrix, you have to first you have to find all of these things that absolutely cannot match at all. At least if you wanted to apply it, right? So by then finding regions in the backwards that are marked, essentially that's what you do, right?
And then what I mean is, like, if you have like 2 matching and 3 non-matching strings, have one kind of potential alignment, it's probably better than if you have like 2 matching and 20
we get into that. I will not repeat your question. Because we're getting into this moment so essentially the question is about two matching. Well, I'll get back to the question in a moment. Ultimately, I already on my examples have put in insertions, also referred to as gaps. You can see that if you sort of want to establish the highest similarity between these two, then this would be the answer. If you allowed for insertions, then this one here would give you higher identity. Right? You just put in something and you can sort of have a high, yes?
Is there only one gap, or could it be?
In this particular case, there is one gap with two amino acids. Could it be anything else? Yes. So, and we get back to that in a moment. In terms of the matrix here, a gap is a move in that direction. That essentially is all. So this matrix idea completely allows you, the dynamic programming allows you to introduce gaps. And you can easily do that. Now, this brings it to, there is this issue. How do I sort of score for or penalize for a gap? We have some penalty to pay that you have a gap. And now the question in this particular case here, we have 2 examples of an alignment. In one, you essentially have one long gap, and in the other one, you have a lot of gaps. But each of these gaps is very short, so essentially only one residue. That brings us essentially to that question that you have. When we look at families, we see blocks. We see that if that is my protein one species, that one another, and maybe there's a bunch of proteins that have something here, maybe a bunch of proteins that have something here, and then there is sort of a gap. Because, for instance, one interpretation of that I said in the last lecture would be that is domain 1 and domain 2. And that, in fact, these 2 species have both domains together, while these species have them maybe in 2 different proteins. And you see that the natural thing then would be, rather than having residue, gap, residue, gap, residue, you would have a long gap. Few long gaps. How can you achieve in the idea of the dynamic programming? How can you enforce that there will be long gaps favored over short gaps? and I made a mistake. I did not really put the score of the matrix that you optimize. In this particular case here really is the identity of the two amino acids minus the gap, the penalty that you pay for the gap. So how can you enforce that you have long gaps rather than many short? Rather one gap of 5 than 5 gaps of 1. How could you put that into the formula? Yeah?
Perhaps in the cost function you can add a factor which is exponentially increasing when your gaps are exponentially increasing with the length with the number of gaps or with the length of the gaps.
So we could have a cost function, she says, that is exponentially increasing with the number of gaps. So the more gaps you put, the more expensive it becomes to do the gaps. Downside, so exponential is always a function that is very, very, very tricky. Be careful when you use the term exponential. So if you have a very long protein, right, then this exponential is going to hit you at some point. Only because you're long, that's unfair. But the idea essentially is completely correct. So you have a way to do it differently, and here it's done in a linear way. You, for instance, have a gap open and a gap extension penalty. And if the gap open penalty is 10 times of the gap extension, then you pay much less to extend the gap than to open it. That is essentially doing it, and this way you get these typical alignments that we have, as it is here. Now, this is for the local version, the paper that published it is from Temple Smith and Mike Waterman, published in '81. Now, ultimately, this brings us back to another question that was asked here. What is better, local or global? So we have these two examples here, and I could possibly have taken the example such that I exclude this gap and that both have an equal number of gaps. But ultimately, the question is, what is the best criterion for matching? What is the best thing in order to sort of assess gaps? I showed you the objective function, and the dynamic programming optimizes this objective function. But is that really the best thing that we could do in terms of biology? And clearly, the best thing cannot be the number of matches. That is clear, because then immediately you would penalize short proteins. It must be a percentage, and even in that percentage thing, it is unclear what to do. But let's look at a most extreme example. Is it better to have 2 hits that are completely right? So all I match between these 2 proteins here are 2 amino acids, these 2. But where I match in this local thing, they're 100% identical, as opposed to a situation where you sort of look at a larger region here, and in this larger region you have, I cannot, you guys are faster than I, 7 by 4, right? No, 7 by 4, exactly. 4 by 7. So it's not 100%. So which one is better? 2 in 2 or 4 in 7? What is better?
So why would you introduce 2 single gaps even though we established the penalties higher?
Okay, forget about the gap. Forget my examples and my images have all kinds of problems. But I could clearly construct, you can imagine that I can construct an example where the gap is not the issue. I just stumbled over the gaps here, that's true. But ultimately the problem doesn't go away. The problem is...
Get to 4 and 7 in the lower one?
Say again?
How do you get to 4 and 7?
Is that another mistake? 1, 2, 3. On the slide before it was 4. 1, 2, I dropped something. 1, 2, I counted 4. Why did I count 4? It's 3. So I made some mistake. So I have to switch some letters here. Let me assume that thank you very much. I hope that I remember to change that. This slide has been with me for 5 lectures now, and this one was not spotted by anybody. And I even counted the 4 in the other one. Anyway, it's totally wrong. So this is 3 and 7. But let's assume this really would be the 4 and 7. Or we could actually even have 3 and 7. The question is the same. Well, 4 and 7 is sort of more appealing as a 3 and 7. You may immediately say, well, 2 is the same as 3, and 2 is better than 3 and 7. With 4, this story no longer is as clear, right? Yeah.
So I would assume that we penalize the more, like the lower the percentage gets, the worse it is. But I would square the percentage so it's as a higher reward when you have a total match of 100%.
In that particular case, then, so he says, I would square the percentage, which means that essentially, in squaring the percentage, you would tend to choose very short ones. So you would argue this one is better than that,
Yeah,
right?
I mean, in the global scale, because when you have a protein that's very short, you have a 100% match, then it's very good.
Certainly true. He says if I think again about this issue of domains, if I match an entire domain entirely, then this is really important. And that is clearly true. But the question here is a more generic one. How can I decide? And the answer is not ask the biologist, because the biologist, well, in this particular case now there's a lot of experience in this field and people do have some answers to that. But what could you do if there would not be a biologist out there who could help you? What would you do?
So a lot of shorter matches make less sense in a way that it's more likely to have two random amino acids just aligned.
Thank you.
Very unlikely to have a long sequence.
So the major point that he said, it makes less sense was the argument, and the reason for sense is it is randomly possible to create that kind of overlap. You have 150 million proteins. Proteins are, imagine they have 500 residues each. So you have 500 times 150 million. What's the probability that you find a particular tumor, a particular set of 2, actually even a particular set of 7 that are identical between any of these? In this huge space, the probability of randomly matching things that are relatively short is very high. So before you can actually answer this question, you have to compute or exploit what's the random probability. If you created some random sets of 7, some random sequences of 7, what's the probability that this particular match, okay, in this particular, now we can go back to 3. So this particular match of GQL, what's the probability that in 7 you match those 3? So you have to assess the background probability. And the background probability will give you the answer to that. Right? That ultimately is the thing. Now, here's another question. So far we spoke about identity. Does that strike you as the most reasonable thing? What else could you look at? Or why may it not be the most reasonable strategy or the optimal strategy to really optimize the identity of letters for the purpose of finding the similarity between two proteins? What could be a reason to look at something else? So there always is the reason that it's so trivial. No, yeah?
Maybe 2 amino acids lead to a similar structure with a higher probability than others.
Yes, exactly. That's the background. So in a more generic way of answering this question, so amino acids, remember I told you, have different features. So some of them are, for instance, positively charged. Some are hydrophobic. And hydrophobic residues tend to sort of cluster inside of proteins, meaning, as you correctly observed, they lead to similar structures. So hydrophobic residues are more similar to each other than hydrophobic and hydrophilic residues are. So matching hydrophobic is more relevant. If you mismatch 2 letters, as long as they're both hydrophobic, it's not as bad as one of them is hydrophilic and one of them is hydrophobic, all right? So you should, and here's an example for that. In this example, this is an important channel protein. The channels are exactly the gatekeepers to the membrane. So essentially they're holes in the membrane. They let things through. This is a potassium channel, so it selectively lets only potassium ions into the cell. And when we look at two potassium channels between two different species, we see the alignment here, and we see that essentially there are three major residues. Doing the structure for the potassium channel gave a Nobel Prize, and in this Nobel Prize it was absolutely essential that the experimentalists realized that these two sequences will lead to the same structure before they knew the structure. It was highly non-trivial. And in fact, they realized that because they saw what's shown here in yellow would be similar. And as you can observe, what is shown in yellow is not always the same amino acid. So only here it's the same amino acid. In these two cases it's not. But it's a sort of corresponding amino acid. And these yellow ones are so important because this is essentially the part of the gate that decides whether it is in potassium or another ion that is let through. Now, yeah?
I'm so sorry, what is the middle line about? Like between pure and saturated?
So that essentially the middle line picks up the matches and into the matches writes some things where, so one way of saying match is identity. That's when the letter comes up here. The plus sign means highly similar. And as you can see in the yellow ones here, this one doesn't even have a plus sign. So there's some sort of biophysical, biochemical similarity between the A and the I, but it's not good enough for the algorithm to pick up, say, that is really similar enough. Yes?
Aren't we here not already in the space where kind of you have to look at the 3D model?
So in this particular case, in order to understand what I'm showing you, he says, are we not in the realm where in order to understand what I'm seeing, I really need to know the 3D structure? Well, if I have to determine the structure of this thing, then no. So the whole thing started exactly here. That led to a structure, that led to a Nobel Prize. But in the beginning, no. So it is about reading sequences. But now the question again is, how would I account for that? How would I build a matrix that compares proteins using some biophysical feature rather than amino acid identity? How could I do this mathematically? How would you create such a matrix? So you could have a similarity matrix, yes, but the question is, how do you create such a similarity matrix? Based on biophysical properties, right, but how do you do that? And you have a bunch of biophysical properties: similarity in charge, similarity in hydrophobicity, similarity in polarity, and then what? Similarity in size. You name all of these features, and then what? Yeah? So that's the essential idea. And that goes back to what the Hennikovs did, Stephen Hennikoff, a while ago in the Blossom matrix. So they look at a set of proteins for which they know they are similar. And then they say if they have a similar function, they have a similar structure, then that means whatever change I observe is not disrupting structure. It's neutral with respect to structure. It's actually the kind of thing that if I align the two proteins, it should be acceptable as a mismatch, and it should be very, very important if it is a match. So essentially they build a log-odds matrix, use this log-odd matrix to align. Let me just show you a few examples of what that implies. Here's the diagonal. And the first thing you see is that the values on the diagonal are not the same for all amino acids. So whether you match a W on a W, or you match an alanine or an A on an A, gives you a very different score. And in fact, by a factor of 8 different score, very different score, right? This essentially implies that some residues are very important to match, and they're very important for the structure, the W. And for some, it is less important even to match them. That's the first observation. The second observation is, when you look at the L matching the M here, you see that L and M gives the same score as the E on the E, and in fact a higher score than alanine on alanine. So this is a mismatch, and what I talked about before, but the mismatch actually gives you more, adds more to your optimum function as a mismatch, this particular mismatch. So again, this is something where evolutionarily or biophysically you can accept this change. And then another aspect here, if you align the A and the I, you actually have to pay for that. So it's an actual cost for this. Today, this matrix here happens to be called Blossom 62. Because of a variety of reasons, and there are many, many, many more matrices out there. Yeah?
Is there alignment where you kind of give a score for two that is matching, or is it only like one on one?
So far, so the question is, could we, so far I talked about alignment being something that does I and J, so that it looks at single residues. So that compares two protein sequences based on the assumption that what happens at position I and I+1 is independent. We know in biology that is not the case. And the question is, are there methods that in fact look at pairs, for instance? So group I and I+1. As pairs, that exists. As things that look at segments, they exist too, but this is immediately getting more complicated. And the price you essentially pay is you can no longer guarantee that the solution you find is optimal. I will briefly run through a few examples next week on Tuesday. Now, the major issue, however, is time. The time, this is square of the length of the protein. Now you may argue, okay, if I have a protein that typically has 1,000 residues, 1,000 times 1,000 is still only 1,000,000 comparisons. It's not that much. You can do that very quickly. Why is that an issue? Why would that be an issue? Because computational biologists don't have computers. Well, there is a little bit of truth to that, but that is not quite the answer. Google has better computers, that's true in Facebook too. But what is the issue here? Yeah?
Because there are much larger layers of input.
So, because we have proteins that are 30,000, but 30,000 times 30,000 still is still a very limited number. You still, in terms of the computer, can easily do that, yeah?
Because you have many thousand proteins.
That is the issue of the database. The major issue here, and let's just go immediately for the crucial thing. Say, for instance, between 2009 and 2014, the number of proteins in the database increased from 8 million to 55 million, which is sort of like computers, right? It's a little bit faster growth than computers, but the number of comparisons that you have to do, you see here is a 20-fold increase every 2 years. So essentially that means that every year this problem is getting harder. And that in fact is exactly the reality of computational biology. The computers are way, way behind the growth of biodata, yes?
But do we actually interested in every comparison between each pair of proteins?
So, are we really interested in the comparison of each pair of proteins? No. The trouble is, before we have seen them all, we don't know which one we are interested in. That's the real problem of the game. Yes, and ultimately you also, with your question, you're hitting exactly in the direction of what we do. This is reality. And the reality is every year getting worse. So now there are two ways of reacting to that as a computational biologist. You can switch the field, you can stop doing it, run away, or you can find hacks. And this is what you essentially say, let me call it a hack. So you find ways of faster comparisons, and part of that we go back into a minute. But you essentially try to find ways, or find out where you don't have to look in order to speed it up through a hack, yes?
But even like 10 to the 12 comparison is not that bad, right? Like in the modern graphic cards, they can give you whatever 8 teraflops. You need like 3,000 tera.
Well, so another way of saying this is that the data is growing too. And this is sort of the last point that I'm trying to make here. When you, in a 5 terabyte disk in 2009, you could put into the plane as a carry-on. It's not funny, it's really true. I took you took all our data from our group that was about that size, indeed as a carry-on, because I didn't want to sort of send it. I also sent it, but I also actually put it as a carry-on in the plane. A few years later, it would have been 300 terabytes, and we cannot do that today. Right? Even today, you cannot carry on 300 terabytes. So even the amount of data that you create with this is something that is more difficult to handle today. But let's just fast forward. How can we speed up the comparison? So one is hack, that we say we don't look somewhere. But how could we really speed up the comparison? And there were some ideas already floating around in some of the questions that go in that direction, yes?
Maybe try to write some hash function that will shorten up the length.
Exactly, some sort of hashing idea. And this essentially is the fast and dirty, and one way of saying that, or one way of explaining that is this one. Assume you began with word size of 3. So here, one idea was the word size of 2. Let's think about a word size of 3, and you simply look at the word 3. Do you find identical match? Yes or no? Okay? Those are the seeds in red here. Once you have these seeds, what you do is you try to extend. At every point where the seed is, you see whether there is an alignment that can extend it in either direction. And then sort of in between all of these here, you can't say anything. Okay? Now, the major issue now becomes one of statistics. Because now you actually have short words. 150 million times 500, I'm totally 10 what is it, 10 by the 10. So in 10 by the 10, the probability, you have 20 amino acids, you have 10 by the 10, so 10 billion residues in today's database, and you have 20 different letters. So the probability that you have all possible pairs of 3, 20 times 20 times 20, is a very, very, very small number compared to 10 billion. Okay? So the probability that you match any word is very high. Okay? And this is why the statistics of distinguishing whether the hit that you have is a random hit or meaningful hit becomes a challenge. It goes back to this question, is 2 out of 2 better than 4 or 3 out of 7? So essentially what you want to do is, you want to, if your method works, you want to have a score that is meaningful, that is very far separated, very far from the background. So the background of random hits is somewhere here, and you hope that the score that your alignment produces is so substantially different from that it cannot happen at random. And that brings us into the next question here: how accurate are pairwise alignments? When does this, I want to compare proteins because I want to figure out whether they have a similar structure. How easy is that actually? Can I figure out that they have a similar structure? Well, there is sort of what I refer to as a daylight zone. So there's a realm, and what I show on this axis is percentage sequence identity. We understand now that what you need is sort of a blossom, a similarity matrix or something like that. But for simplicity, I'm going to continue to talk about percentage sequence identity, so identity of letters. If 2 proteins have 100% identity, it's clear they have a similar structure. Okay? So there's somewhere it's clear that the problem is trivial. And then, you know, the more you lose of the similarity, the more the problem becomes one that is no longer as trivial. But the part where there is essentially no mistake done is what I call the daylight zone. So you don't make a mistake, essentially the alignment is easy. And that may be down to levels round about 40%, which actually is a large realm, but that's true. Then we get into a zone that Russell Doolittle coined the term of twilight zone. So that's where you sort of lose the signal. You can no longer clearly say there is a signal or not. And then you continue going into the midnight zone. And essentially the idea of a midnight zone really is that at this, when you enter that, the probability of a random hit is as good as the probability of the hit that you have. So you can no longer distinguish between a real similarity and one that has happened by random. Now, yes, sorry?
One question, the X scale, what would be the unit
In this particular case, the unit really is percentage sequence identity. So, and this is not misleading, this is not quite true, but the idea that if you have two proteins that have 40 or 50% sequence identity, so 50% of their amino acids have the same letter, then essentially they have the same structure. Okay? And now my question is, if that is true, and then there's this twilight zone, how rapid is the collapse, so to speak, going to be in the twilight zone? And clearly on the other end is also very simple. If the percentage sequence identity is 5%, that's random. We have 20 amino acids, 100 by 20 is 5. So this explains, I'll pick it to 10 here. So this number here is 5%, right? So this is clearly random. The midnight zone that I show here starts earlier. And that's roughly also true. It's a sketch, but this is not that wrong. Now I'm going to show you, I want to sort of study how does similarity in structure, according to divergence, change from I'm always having the same structure to a point where I sort of almost never have the same structure. And what's the transition going to be like? And I'm going to show you graphs that look like this, where again the x-axis here is percentage sequence identity, and then I'm going to have number of pairs here, number of pairs that have a different three-dimensional structure, and number of pairs that have the same three-dimensional structure. This is the sort of thing that I want to show. Okay? How can I do that? How can I produce that? If you had to do that as an exercise, how would you produce the data? And I'm not saying Google and find some data set that you can plot. I mean, how would you produce the data set? Yes?
What are the axes again?
So this axis essentially, you have a pair of proteins and you are essentially asked how many amino acids are identical between these two in the optimal superposition. On that axis, you simply have a number of pairs of this. Red, pairs of proteins that have a different structure. Blue, pairs of proteins that have a similar structure. Yeah?
Would it better just to visualize it as an ROC curve?
We will get to ROC curves. So the question is, would it be not better to have an ROC curve? This is a debatable, I would argue for the beginning not, but this is not really addressing the question how to create the data. How to visualize is a different story. No, I believe there's a good point to start like this. Because by getting numbers, you get some impacts. I'll show you in a minute why this brings you somewhere. Ultimately, you will want to see rock curves, and you will see them. But the question really is, how can we do that? How can we create the data? How can we... what I showed you before was not a real plot. It's just you want something that looks like that. How can you create the data? What do you need?
Similarity and probability. We can use similarity on one axis and probability to the candidate based on.
So we use sequence similarity. So you need a bunch of alignments, yes. You need a bunch of proteins that you can, for which you can compare their sequences, yes. And then what?
And then on the y-axis, maybe we can take the probability of them being similar.
No, so in the y-axis for the time being is not really properties, it's just numbers. But my question is, how do I create red and blue? And the answer in this particular case is not use a different color. This is not what I mean. What do I have to do? What do I have to give you? What will, if you were doing this exercise, what would you be asking me for? Yes?
To plot the numbers, I need to see the structure.
You need to see the structure, yes. So we want to play this on several thousands. We want to play this on as many as possible. So seeing the structure, I need structures. That is certainly true in what you say.
I need a database.
A database of structures. So I give you a database of structures. Now what? Yes?
If you have a database with the proteins and labels corresponding to structures, then you can, or even if you have full information on the sequence,
You're on track. You're on the track, but you haven't come there right. There are a few details that you need to cut into. So again, yes, I have the three-dimensional structures. I can look at the sequence comparisons. Yes, and I need labels. Where exactly do these labels come from? Nobody's going to give them. I tell you that you already have them, or you can produce them with the tools that we have already discussed. How? I say I give you 100,000 proteins of known three-dimensional structure. I give you a tool to compare proteins on the level of the sequence and on the level of structure. So 2 tools. What can you do? Yeah?
We can probably generate a number of test sets just by, well, running the tools completely, not on the modified sequences, and then use this as a benchmark.
Yes, but I want more detail. You say I can create my benchmark. Yes, but how? You can run the tools and create your benchmark. True. So if we would be in an exam, and I would say this is nothing you answered to my question is wrong. But you didn't answer my question. How? How do you exactly use which tool to do what?
You use probably the tool that looks for the whole structure for the benchmark, because it's more computationally hard, but it provides answers that are guaranteed to be true.
Yes, but what do you do with that? So you have a tool that looks at structures, and what do you do?
Correlation between sequence similarity and structure similarity?
Yes, so that's exactly the crucial point. Now, you have to have set of pairs. And for each set of pair, you have to create a sequence similarity and a structure similarity. You have to compare them on the level of sequence, and you have to compare them on the level of structure. Essentially what you do is you do the entire database of protein structures. You do a 3D comparison and do a 1D comparison. And those are the 2 pieces of information you want to correlate, okay? You have to do that. And then you're going to create some distinction that you say if it is more similar by some root mean square deviation than something, I call it similar or same. So that's the blue. If it is more different than something, I call it different, and paint it red. And then there's a large thing in between where I will say I will ignore that. There's sort of cases in between. I want a very clear-cut answer, so I'm going to ignore all of these cases in between. Okay? So now I have red and blue. There is some additional problem here. All biosets are, because of a variety of reasons, some of those are related to evolution, highly biased. Biased means there's many repetitions of the same thing. PDB has about 150,000 proteins in there. If I just take the first and want to build a unique data set, unique meaning that no pair has exactly the same sequence, then what is left is only half of it. For half of all the entries of the PDB, I have the same twice. There's a variety of biological reasons why that is the case. But that creates, in my counts here, that creates something that is absolutely not reflecting truth. It's reflecting some bias in the data set. So that means that we have to sort of look at some unbiased subset. So I have to pick out a set from the PDB that is such that no pair in there is more sequence similar than something. So they are not trivially similar. And then I sort of compare this against everything else. And then when you do that, you create something that Chrysander and Reinhard Schneider, he's not at Harvard University, originally publishes the HSSP curves that essentially show number of residues aligned versus percentage sequence identity. So the axes from the plot I showed you before are inverted. So this is again now identity in terms of 1D. Same letter, 100% means that all the letters are identical that I align. And this is number of residues aligned. That curve is such that blue is above, red is below. The curves changed a little bit later, and here's sort of the underlying data. Blue, pairs of proteins that have a similar structure. Red, pairs of proteins that do not have a similar structure. You see that the line, the gray line here somehow distinguishes, although there's a little bit of red above. But clearly most of what you see up here is blue. There's also some blue below, and what I'm really not showing you is the density. In order to get the density, we would essentially have to sort of look. What we're going to, on the next plots, what I'm going to show you is what is the sum over all these points above the line. And the sum is going to, as an axis on the x-axis, what I'm going to use is the distance to that gray line. So in the distance to the gray line, this is the gray line of 0, plus 5, plus 10, and so forth, minus 5, minus 10, and so forth. Right? This is what I'm going to show you. So that is this what I'm saying here. 10, 5, 0, minus 5, minus 10. And it corresponds again to something that you could say is 20% here. This axis is the number of pairs on a logarithmic scale in blue. And you see, the first thing that you observe is that upon this entering this line, this gray line that I showed you before, the number of pairs that you find that have similar structure explodes. Exploding, again, it's more than a factor of 10, right? So going from 5, plus 5 to minus 5, you have 10 times, or you increase the number of finds, the number of similar proteins by a factor of 10, right? So this means that, and that ultimately is the twilight zone, right? This means that we should do our alignment as far as we can. But there is a price to pay. Now I have to show you the red. And that is the red. So we begin from somewhere where we essentially make no mistakes over the entire data set that I have, to some place where most of the things that we find are wrong. So let's just pick this point here. In this point, more than 90% of all my findings at minus 5 here are red. More than 90% of what I find will be mistakes, okay? And here, essentially 100% are right. Now, that is what I mean by an immense transition. This is essentially what physicists call phase transition. You go in a relatively small number of steps of percentage sequence identity, and you go from a problem that is totally trivial to a problem that you can actually not handle. And you have to do that because most of the treasure that you want to find, most of the hits you find, are exactly in the region where it gets difficult. Okay? That's the challenge and of database comparison, of sequence comparisons. And this challenge essentially is addressed through multiple sequence alignments. And well, I can push a little bit further. So the first idea could be that we do multiple sequence alignment through dynamic programming. The problem really is, since it's a problem that has to do with the square of the length, we sort of now get it into the cube of the length. And we do have an issue. So today there seems to be solutions possible for up to 6, and I have not found the quote here, but somebody told me that somebody had programmed it, and so they used 60 terabytes in order to align 6 proteins. It's not so scary. It is already a fairly scary thing, but the fact is that is 6. Typical protein families have many thousands of members. So we clearly cannot handle protein families as we know them. There's a hack. And essentially the hack is sort of looking at something in the vicinity of the interaction, and that ultimately goes, the other hack is map it to a tree, and that is the one that I want to follow up on. Again, from Rost, the one who coined the term twilight zone. The idea is very simple. say you had 4 proteins to compare. You compare first pairs. You do A against B, A against C, A against D. And that establishes a pairwise similarity. Now what you're going to do in the next step is, first you're going to align, say, A and B. And this gives you this alignment, or let me just show you the alignment in step 1. So the alignment would be this one here, where the small letter underneath here is the consensus, meaning in this particular case here, wherever it's the same residue, I take that residue, and where it is a different residue, such as in this case here, I take the one from the first. So I create a new sequence. That is sort of a merger of these two. Once I have this new sequence, that sequence represents the information that I gained from aligning the first two. I do the same thing here with the second pair. So the second most similar, so I begin with the first pair being the one that is at the highest sequence similarity, and I take the second most high that doesn't overlap, which would be C and D. Create another one of these sort of consensus sequences, and in my third step, I simply take these two consensus sequences and align them. There's a simple way to do 4 sequences aligned build them up through pairwise alignments. Right? The idea is clear, or not? Is there anything unclear about the idea? Yeah?
Don't you just pretty much always get that first sequence again?
So, okay, the way I construct my alignment, yes. Okay, let me change the algorithm now such that what I put in here is not the first, but I do the one that is most surprising. Okay, so I use a matrix, the Blossom matrix, and then I would have a mechanism by which the most surprising letter at that level could be the D, and then I would no longer get the first. Yeah? And I thank you for that. So sometimes by simplifying issues, you make a mistake in simplifying. So the way I described the algorithm, you absolutely correctly spotted that, was such that if you had... So those of you who accepted what I said, they actually accepted that you just... The alternative algorithm would have been align these two by taking this, align these two by taking this, and then essentially only align these first two. Which means that you, instead of compare 4 sequences, you only compare 2. And that ultimately is what I sort of sold you underneath in my unclarity of speaking, and you spotted that. Thank you. But again, the consensus sequence I could redefine, so I should change my example here and put a D in this particular case. And then where it's different here, in this particular case an L. So just to show that there is some sort of random thing that I'm not actually creating, pick sequence 1.
Kind of random mergers of these?
So then it's not a random. So the way you actually do it is by looking at the information content. So you pick the residue that at that particular position is the most informative evidence. And that really has to do, if it's an A, you already saw in this Blossom matrix, matching A on A is sort of irrelevant, so you would not take the A. I'm not sure whether I have an example where the A disagrees. No, there's no example where the A disagrees. But if the A disagrees, one is an A, one is an E, you take the E. Okay? Because the E has more value, more information content. Yes?
So step 3 is just to put the 2 merge words next to each other, but what do you do? Do you match them, and where do you put the match number, and what do you control?
So what do I actually create when I have 3? So upon having created 3, I create, so the way to think about it is you create a new sequence, and what do you do with the sequence? Well, that is sort of a misleading way of presenting it. I only wanted to explain how you can map the problem onto something that is very easy to understand. And what you actually do with it is you now know how to put all 4 underneath each other. So this sequence that you create from this process is irrelevant. What is relevant is the way you align. Is that a clear statement or not? It's not clear. So what I mean is that I have to find a different example.
So what I thought we were trying
No, my problem really is the example. So I need examples where things move around. In the examples that I create, things are too similar. So ultimately where you're going is you want to create this alignment here. You want to create the alignment between those 4 sequences. You want to see whether the 4 sequences are aligned as it is shown here. And in this particular case, the case is so simple and it's trivial. There's no other way of doing it. But you could imagine that the last step is not creating another consensus sequence, because that is meaningless, but is actually creating a way of readjusting the alignment between these 2 and these 2, moving it 1 over, for instance.
Okay, so we did dynamic programming to show the similarity matrix, but with the similarity of the 4, there's nothing we could make it. We just have similarity of 4 elements, which was previously not what we tried to get. We tried to get similarity between 2 separate elements.
Yes, so this is true. We are not, the meaning of a hack is I'm not solving the problem of really getting the optimal similarity between all 4. Hack means I reduce the problem of finding the optimal similarity between all 4 to many pairwise similarities. Because pairwise similarities I can solve with the dynamic programming. And I cannot expand the dynamic programming at all to the problem of 4. Well, again, as I said, in the computer I can do 6, but I cannot do 1,000. And therefore, but I can easily map, if I want to, instead of having 4 here, I had 1,000, I can do pairwise 999 times 1,000 divided by 2, 999 by 500. So that I can relatively easily do. As long as I can sort of map it to the pairwise alignments. And again, so the answer to your question, what is the result of that? The alignment that you see on the whiteboard. And this allows you, this multiple sequence alignment, to sort of intrude into the twilight zone. There is a slightly different way of doing that, but ultimately that is the simple idea behind it. I'm going